Welcome, poultry and genetics enthusiasts. You're about to dive into Chapter 6 of our basic poultry genetics course. If you've missed previous chapters, don't worry, you'll find all the links in the description to catch up and delve deeper into this fascinating world. Today we're going to tackle those questions that pique many of our curiosities. Why do some chickens have red earlobes and others white? Is there a secret connection to egg color, from classic whites and browns to astonishing blues and greens? And what about that old myth linking earlobe color to the number of eggs a hen lays? Is it possible that genetics holds the keys to understanding it all? I'm sure it does, and what do you think? Get ready for an epic journey into the heart of these incredible birds' DNA. We'll uncover how science has unraveled the biochemical mechanisms that dye their earlobes and shells, the truth behind the genetic linkage that creates those curious correlations, and why egg production is a much more complex and surprisingly independent trait than you might imagine. We'll talk about genes with fascinating names, genes that change colors, and how human selection has shaped the chickens we know today. It's a story of science, mystery, and demystification you won't want to miss. Let's begin. When we observe chickens, we immediately notice that some have red earlobes and others have white. This seemingly simple characteristic has a clear genetic basis. Most of the time, we're talking about a monogenic trait, meaning a single main gene or a set of very closely linked genes controls it. Earlobe color, like feather or skin color, is due to the presence or absence of melanins, which are the body's natural pigments. The primary culprit behind the reddish and yellowish tones in our chickens' earlobes is pheomelanin. This is synthesized in specialized cells called melanocytes, found in the earlobe tissue. If a hen's genes instruct these melanocytes to produce and deposit pheomelanin, we'll see those characteristic red earlobes. Imagine a small pigment factory operating right there, painting our beautiful chickens' earlobes with lovely red or yellowish hues. In contrast, white earlobes simply lack pigment. This can happen because melanocytes aren't present in that specific area, or because they aren't functional and can't produce melanins. It's as if the pigment factory is closed in that part of the body. Amazing, right? Although there isn't a single earlobe color gene with a specific name, science points to genes that regulate the formation, migration, and activity of melanocytes. Genes like MITF, melanocyte-inducing transcription factor, or TYR tyrosinase, which are crucial for overall pigmentation, could indirectly influence whether pigment is expressed in the earlobes. Exceptions to the rule? Of course there are. Some breeds, like the exotic Silkie bantam or certain specimens of Vietnam's Dong Tao breed, dragon chicken, might surprise you with bluish or purplish earlobes. But here's a trick. It's not a blue pigment. This color is the result of an optical effect called light scattering, similar to how the sky appears blue. It's due to layers of densely pigmented connective tissue with dark eumelanin beneath a translucent epidermis. This is a manifestation of fibromelanosis, a genetic trait that causes widespread hyperpigmentation of connective tissues in these breeds. We can also find cream or yellowish earlobes, suggesting a lower concentration of pigment or the influence of other modifying genes. On the other hand, eggshell color is a true palette of nature, and its genetics are fascinating. Unlike earlobe color, this trait is predominantly polygenic, meaning many genes work together to produce the final color. Shell pigments are deposited in the hen's uterine gland, or shell gland, within the oviduct, just in the final hours before laying. It's an astonishing process. For example, white eggs are the default expression if no other pigments are deposited. Genetically speaking, they are considered recessive to colored eggs. The White Leghorn is a classic example of a breed fixed for this color, being one of the world's most efficient white egg layers, along with other well-known breeds, generally of European origin. The commonly brown eggs we find in supermarkets, or even dark brown ones, feature protoporphyrin as the star pigment. This compound is a byproduct of heme metabolism, which gives blood its color. It's synthesized in the uterine gland cells and deposited on the outer surface of the shell in its cuticle. That's why if you scratch a brown egg, you'll see the white shell underneath. The intensity of the brown color varies enormously, from pale creams to intense, almost chocolate browns, because many genes influence the amount of protoporphyrin produced and deposited. It's a quantitative trait, meaning it's affected by the accumulation of multiple genetic effects and, especially, the hen's diet. Protoporphyrin is formed from common precursors in the heme pathway. In the uterine gland cells, this pathway is diverted to produce free protoporphyrin, which is then secreted and adheres to the shell. The activity of key enzymes in this pathway, such as ferrochelatase, though better known for incorporating iron into heme, its regulation can affect the availability of porphyrins. And membrane transporters in the gland's epithelial cells are crucial for the amount of pigment deposited. It's like a faucet that's open more or less, controlling the flow of brown dye. Interesting, right? I find the genetics in our chickens fascinating. In the case of blue or green eggs, the main player is biliverdin, often in the form of a zinc chelate. Biliverdin is a product of heme degradation. Unlike protoporphyrin, biliverdin is incorporated directly into the calcium carbonate matrix of the shell as it forms. This means a blue egg will be blue inside and out. Consequently, green eggs are a mix. They occur when a hen that genetically produces blue pigments, biliverdin, also has the ability to deposit brown pigments, protoporphyrin. The combination of blue and brown creates that greenish hue. Incredible, right? Biliverdin is primarily produced through the enzyme heme oxygenase, which degrades heme. What's fascinating in blue egg-laying chickens is how the activity of this enzyme is regulated in the uterine gland. Once formed, biliverdin is transported and binds to the calcium of the shell as it calcifies. What genes are behind this? For blue eggs, we have the famous Oocyan gene. This gene has been found not to be a normal gene, but rather an insertion of an endogenous retrovirus in the regulatory region of a gene called SLC30A7, which is a zinc transporter. This viral insertion, still under study, somehow switches on the machinery for the shell gland to produce and deposit biliverdin. Breeds like the Araucana and Ameraucana carry this fascinating gene. The insertion of this endogenous retrovirus is an extraordinary genetic event. Retroviruses, like this one, insert their genetic material into the host's DNA. When this occurs in an animal's germ cells, the insertion can be inherited for generations. In the case of the Oocyan gene, the insertion happened at a key location near the SLC30A7 gene. Although the precise function of SLC30A7 in shell pigmentation is still under investigation, it's known to be a zinc transporter, and zinc is a component of the biliverdin chelate. It's believed that this viral insertion altered the regulation of SLC30A7 or nearby genes, leading to the activation of the biliverdin pathway in the uterine gland. It's an astonishing example of how an accidental event can lead to such a distinctive trait. For brown eggs, the genetics are more complex. Multiple quantitative trait loci, or simply QTLs, have been identified on various chromosomes. These QTLs contain genes that likely influence the efficiency of protoporphyrin synthesis, its transport, and the time the egg spends in the uterine gland, which directly affects the amount of pigment deposited. The wide variability in brown shades is because the dose of pigment is controlled by many small genes. Some identified QTLs are associated with enzymatic activity in the protoporphyrin pathway, while others may influence the speed of egg transit through the uterus. More time, more pigment. It's like having multiple small switches that control the intensity of a light. The combined effect of all of them determines whether the egg is a pale cream or an intense chocolate brown. In addition to genetics, factors such as stress, the hen's age, or certain diseases can cause the shell color to temporarily become paler. For example, an older hen might lay lighter brown eggs than when she was young. This isn't a genetic change, but a variation in expression due to external or physiological factors. Knowing all this, we arrive at the million-dollar question, the observation that has guided breeders for centuries. Why do chickens with red earlobes often lay brown eggs, and those with white earlobes lay white eggs? This is a very strong empirical correlation, a golden rule in poultry farming, but it's not a cause and effect relationship. It's not that the earlobe color causes the egg color. The most accepted explanation is genetic linkage. Imagine that the genes for earlobe color and the genes for egg color are very, very close together on the same chromosome. When genes are linked, they are inherited together most of the time, as if they were a package. Historically, breeds that developed red earlobes also carried the genes to deposit protoporphyrin in their eggs, resulting in brown eggs, and these genes remained together through generations due to selection and crossbreeding. The same happened with white earlobed, white egg-laying breeds. Is there a single gene that controls both traits, or does one gene affect the expression of another? While these are fundamental genetic concepts, there's no strong evidence to show that the same biochemical pathway directly controls pigmentation in the earlobes, melanins, and in the eggshell, porphyrins or biliverdin, in a pleiotropic manner. The metabolic pathways for these pigments are different. However, epistasis could play a subtle role in modulating the intensity or expression of certain colors if one gene suppresses the action of another at a key point in pigmentation. This adds layers of complexity to genetic interactions. Exceptions are key to understanding this. The Spanish Penedesenca breed is a perfect example. This breed has white earlobes, but it lays very dark brown eggs. This is irrefutable proof that the genes for earlobe color and egg color are independent in this breed, or that the genetic linkage has been broken. This occurs due to genetic recombination. During gamete formation, chromosomes can exchange segments, separating genes that were linked. Araucanas and Ameraucanas are also an interesting case. They typically have red earlobes, and they lay blue eggs. Here, the Oocyan gene for the blue egg acts independently, without being linked to the earlobe genes in a way that changes their color. These exceptions demonstrate that, while the correlation is very common and useful, it's not universal. Genetic linkage, a result of thousands of years of evolution and artificial selection, is the main reason behind this curious relationship. And now, the big myth debunked. Does earlobe or egg color influence how many eggs a hen lays? The answer is a resounding no. Egg production, or prolificacy, is an incredibly complex genetic trait, what we call a quantitative trait. This means it doesn't depend on a single gene, but on the interaction of hundreds of genes, each with a small effect, plus a strong environmental influence. These genes control everything from ovarian follicle development, hormonal regulation, nutrient metabolism, to disease resistance. The modern high-producing hen is a marvel of genetic selection. The genetic and biochemical pathways for pigment production are entirely different and separate from those controlling ovulation and egg formation. There is no direct physiological link. So why are breeds like the White Leghorn, with white earlobes and white eggs, famous for their extremely high production? This is where artificial selection and co-selection come into play. For decades, even centuries, breeders relentlessly selected chickens that laid more eggs. The Leghorn, for example, already possessed a natural genetic combination that gave it a great laying capacity. By selecting and breeding the best layers, breeders incidentally fixed their earlobe and egg color characteristics as well. They weren't selected for having white earlobes or white eggs, but because they were excellent layers and, coincidentally, had those aesthetic traits. It's a genetic package that proved successful. In short, the number of eggs a hen lays is the result of a complex set of genes related to her reproductive physiology, completely independent of the genes that determine the color of her earlobes or the color of her eggshells. To truly understand the genetics of earlobe and egg color and how it relates to laying ability, we need to talk about the chicken genome. The complete sequencing of the chicken genome has been a scientific revolution. It has allowed us to identify the precise location of many genes on the chicken chromosomes. Thanks to this information, scientists have been able to create detailed genetic maps. This is like having a GPS for genes. It allows us to identify QTLs, quantitative trait loci, which are specific regions of DNA that contain genes influencing complex traits like egg production. Understanding these QTLs is key, because laying ability doesn't depend on a single gene, but on the combined action of many of them, distributed throughout the genome. Sequencing has also helped to refine the search for the exact genes responsible for pigment synthesis and deposition, both in the earlobes, melanins, and in the eggshell, protoporphyrins and biliverdin. Although the Oocyan gene was already identified, the genome helps us better understand its mechanism of action at the molecular level, such as that retroviral insertion that activates blue pigment production. Knowing the genome gives us the tools to understand the genetic architecture of these traits, allowing us to see how the building blocks of DNA construct the complex biology of the hen, and thus we can modify them to our advantage. Imagine for a moment breeding Leghorn chickens with red earlobes that lay lots of brown, blue, or green eggs. Wonderful, right? Or having a dual-purpose hen that lays few eggs compared to commercial lines producing over 320 eggs a year, like an Isa Brown. Anything is possible with genetics. We already mentioned genetic linkage as the primary reason behind the earlobe and egg correlation, but genetics is rarely that simple. Two key concepts that add layers of complexity are epistasis and pleotropy. Imagine an orchestra where one musician, a gene, can silence or change the sound of another musician, another gene. That's epistasis. One gene can mask or modify the expression of another gene in a different trait. For example, in feather color, the eye gene, inhibitor, in some breeds can make a chicken white, even if it genetically carries alleles to be black or red. In the context of eggs, there could be subtle epistatic effects influencing the intensity of brown pigments, where the expression of a gene for protoporphyrin production is affected by another regulatory gene. As for pleotropy, here a single gene has multiple seemingly unrelated phenotypic effects. For example, if a gene affects a metabolic pathway crucial for the synthesis of a type of pigment, and that pigment were used in both the earlobes and the eggshell, we would be talking about pleotropy. Although current evidence doesn't point to direct pleotropy for the earlobe and egg correlation, given the difference in pigments, this concept is fundamental in genetics. A clear example of pleotropy in chickens is the gene that causes fibromelanosis, dark tissue, in the Silkie, which affects the color of the skin, bones, and, as we saw, the bluish earlobes. Understanding how epistasis and pleotropy work helps us see that biology is not a set of simple switches, but an intricate network of interactions. Traditional selection by observation has been powerful, but modern genetics allows us to be much more precise. We're talking about marker-assisted selection, which will result in a bird with the physical and productive traits we are looking for. What are genetic markers? They are specific DNA sequences, like signposts, that are very close to the genes we're interested in. For example, if we know that a particular marker is almost always present in chickens that lay blue eggs or are highly productive, we can try to introduce these characteristics or that marker into very young chicks. Instead of waiting for a hen to mature and start laying eggs to see her production, or to see the color of her eggs, we can take a small DNA sample from a chick and analyze its markers. This allows us to identify the best breeders, males and females, much earlier, drastically shortening selection cycles and accelerating genetic progress in traits like prolificacy or desired egg color. It's like having a genetic crystal ball. We can also use markers to select against undesirable traits, for example those associated with shell fragility, or to avoid excessive inbreeding, maintaining genetic diversity within breeding lines. Marker-assisted selection is the pinnacle of applied genetics in poultry farming, allowing us to genetically design the chickens of the future more efficiently and precisely. Although we are focusing on chickens, the genetic principles governing color and other traits are universal in the animal kingdom. This gives us a broader perspective on the incredible biological diversity. The synthesis of melanins, eumelanin, pheomelanin, is a fundamental biochemical pathway present in almost all animals, from mammals to insects. Understanding how genes control the distribution of these melanins in chickens, in feathers, skin, earlobes, has direct parallels with coat color in dogs or cats, or skin in humans. It's the same basic molecular machinery, just expressed in different ways. Remember the earlobe and egg correlation in chickens? In other species, we also find curious correlations of traits that are often due to genetic linkage or pleotropy. For example, in dogs, certain coat pigmentations like merle are linked to hearing or vision problems. In some fish, coloration patterns are intrinsically linked to aggressiveness or reproductive success. These parallels show us that nature's genetic engineering often groups genes that evolve together or share unexpected functions. Studying these traits in chickens also helps us understand broader evolutionary processes, how natural or artificial selection favors certain packages of genes, and how mutations, like the viral insertion of the Oocyan gene, can lead to completely new and attractive phenotypes. These are many examples of evolution in action. By observing the genetics of color and laying ability in chickens, we are not only learning about birds, but about the fundamental principles that govern life and evolution across the planet. And we've reached the end for today. A deep dive into the amazing genetics that shapes our chickens, from the color of their earlobes to the eggs they gift us every day. We've debunked myths and discovered the true science behind those curious traits. I hope you enjoyed this genetic journey as much as I did. If you found this video fascinating, don't forget to give it a big like, share it with other chicken lovers, and of course subscribe to the channel if you haven't already. And most importantly, hit that notification bell so you don't miss any of our upcoming videos. Plus, I have great news for all animal lovers. We're launching a new channel dedicated exclusively to other varieties of farm animals, where we'll talk about cows, ducks, horses, sheep, and much more. The link to our new channel can be found right below, in the video description. It will be an incredible extension of our community, and we hope to see you there. And for those who want to continue exploring the fascinating world of poultry genetics, remember this is Chapter 6. All links to previous chapters of our basic poultry genetics course are available in the description. To your success, fellow breeder, until next time.